There is dust falling in the room where you are lying tonight. It is falling slowly, far too slowly to see, in the way that dust does when there is no wind and nothing moving, and the light has gone out of the window. Fibers from the blanket and skin, and small fragments of the day, all of it drifting down through the quiet air at a speed you could measure in millimeters, settling on the shelf and the floor and the back of your hand. A soft, continuous fall that does not stop when you close your eyes and does not stop when you sleep. It is one of the gentlest processes there is. Nothing about it asks anything of you. Now hold that in mind and take it out to sea, because there is a place, 600 meters down and more, in the middle water of the warm oceans, where exactly the same thing is happening on a scale that never ends. Everything that lives in the bright surface of the sea eventually stops living, and when it does, it begins to sink. The bodies of animals too small to see, the clear, discarded shells of creatures that build a new house for themselves every day and let the old one drop, the waste of the shrimp-like animals that graze in the shallows, broken diatoms, threads of mucus, dead algae. It all comes apart and comes down, in a fall so slow and so constant that biologists gave it the only name that fits, and called it snow. And somewhere in the middle of that falling, in water so dark that the idea of color has stopped meaning anything, there is a small animal about the length of your forearm, hanging almost perfectly still. It is not swimming. It is not hunting. It has one thin filament extended out into the water, longer than its whole body, held out into the falling like a hand held out into rain, and it is waiting for the snow to land on it. That animal was given a name in 1903, and it was a name about hunger and darkness, and it has been misunderstood for very nearly the entire time since. What it actually does down there was not established until 2012, and when it finally was, it turned out to be the gentlest thing that any cephalopod has ever been observed to do. Welcome back to Sleep Abyssal. Settle in, let the room go quiet, and let the sea take the weight of the day. Tell me in the comments where you are listening from tonight, and whether it is raining where you are, because tonight the whole video is about falling. For the next 2 hours, we are going to sink very slowly out of the sunlight, down through the fading blue, into a layer of the ocean that almost nothing can stay in, and spend a long, unhurried night with the last surviving member of an entire order of animals. We will start with the ship that found it, and the man who named it. We will spend a while looking at it properly, because it has been drawn badly and described badly for a very long time, and then we will follow it down into the dark and stay there, in a slow drift, until you are asleep. If you find it easy to drift off here, a quiet like helps others who are looking for the same calm to find their way to it. And that is all. Now let us go back down. The story begins on a steamship in the summer of 1898, in the harbor at Hamburg, with a great deal of rope. The vessel was called the Valdivia. She was not built for science. She was a working steamer of the Hamburg America line, a cargo and passenger ship of a little over 2,000 tons, and she had spent her life carrying ordinary things across ordinary distances. In 1898, she was chartered by the German government, taken into a yard, and rebuilt inside for a single purpose, which was to find out what lived in the deep sea. That question had a particular shape at the end of the 19th century. 20 years earlier, the British had sent out the Challenger, a converted warship with most of her guns removed, and she had spent 3.5 years going round the world with a rope and a bucket, sounding the depth and dredging the bottom and hauling up, again and again, animals that nobody had ever seen. The Challenger came home in 1876, and the writing up of what she found took the better part of 20 years and filled 50 volumes. It settled, permanently, an argument that had been running for decades about whether anything at all could live below a few hundred fathoms. It could. It did. The floor of the ocean was inhabited everywhere they looked. What the Challenger had not really touched was the water in between. The bottom is one thing. You lower a dredge, it lands, you drag it, you pull it up, and whatever was on the mud comes with it. The middle water is much harder. It is a volume rather than a surface, an enormous three-dimensional space with no floor to catch your net against. And to sample it properly, you need to be able to open a net at one depth, tow it, and close it again before it comes back up through everything above. Otherwise, you never know where anything came from. A creature in your net might have come from 1,000 meters or from 30. The Valdivia carried closing nets, and the men aboard her understood that this was the point. The expedition was led by a professor of zoology from Leipzig named Carl Chun. He was in his mid-40s, careful, methodical, and by all accounts extremely difficult to distract. He had already spent years working on the plankton of the Mediterranean and the Canaries, and he had a particular interest in the animals of the open water, the ones with no rock to hold onto and no burrow to hide in, whose whole existence takes place suspended in a fluid. He had argued, before the ship even sailed, that the deep middle water would turn out to be full of life adapted in ways nobody had guessed. The Valdivia left Hamburg on the last day of July in 1898. She went north first, up towards the Faroes and Iceland, then turned and ran south down the length of the Atlantic, calling at the Canaries and the Cape Verde Islands and the coast of West Africa. She rounded the Cape of Good Hope and went south into the cold, further south than any research vessel had gone in that sector for a very long time, down to the edge of the pack ice. Along the way, she found again a scrap of land called Bouvet Island, which had been sighted in the 1730s and then effectively lost, its position recorded wrongly, so that ships had been sailing past the place it was supposed to be for 150 years without finding anything. The Valdivia put it back on the chart. Then she turned north and east, across the southern Indian Ocean, up past the Seychelles and the Maldives, over to Sumatra, and home the long way. She was at sea for 9 months. She covered something in the region of 32,000 nautical miles. And at station after station, the closing nets went down into the middle water and came back up again. They came back full of things nobody had names for. That is the ordinary condition of deep sea work in that era, and to some extent it still is. The nets brought up transparent animals, animals with organs that glowed, animals with eyes on stalks, and eyes pointed permanently upward and no eyes at all, fish the length of a finger with mouths that unhinged, jellyfish that fell apart the moment they touched the air. Chun collected all of it. He preserved what could be preserved, and he did something that made a real difference to what came afterwards, which was to have the animals drawn and painted quickly, while they still had their colors, before the alcohol took everything away and left it the same tired brown. And somewhere in that hall, in the tropical Atlantic, off the coast of West Africa, came up a small dark animal about the size of a hand. We do not have a good record of the exact moment. What we know is what Chun eventually wrote about it, and what he wrote suggests he took it for one thing at first, and only slowly understood it was something else. It was soft and gelatinous, and it had 8 arms bound together by a web of skin, so that when the arms were closed, the whole thing formed a rounded bell, and when they were opened, it spread into something like an umbrella turned inside out. It had 2 small fins on the body, which octopuses generally do not, though some deep-sea ones do. It had a pair of eyes that were, for the size of the animal, quite extraordinarily large. Its skin, before the preservative reached it, was a dark velvet color, somewhere between deep red and black, and the web between the arms was a purplish shade that some later writers called plum and others called wine. The eyes had a reddish cast. The beak, deep inside the web, was pale. Chun described it as a small finned octopus. He thought it was gelatinous and probably not very active. On both counts, he was closer to right than almost anyone who followed him for the next 50 years. And then he named it. The name he chose was Vampyroteuthis infernalis, which translates directly, without much room for softening, as the vampire squid of hell. That is a striking thing to call an animal, and it deserves a little unpacking, because it has done more to shape how this creature is thought about than any piece of research since. Naming, in the biology of that period, was not the neutral bureaucratic exercise it has largely become. Latin binomials were often descriptive in a frankly poetic way, and a naturalist who had spent 9 months in a laboratory built into the hold of a converted cargo steamer, looking at hundreds of animals nobody had seen, was under a certain pressure to be memorable. Chun was looking at a dark red animal with a cloak of skin between its arms and a pair of deep-set reddish eyes, in the last years of a century that had produced a great deal of literature about cloaks and darkness. The reference is not subtle, and it was not meant to be. He was describing a color scheme and a silhouette. He was not describing behavior. There is nothing in his account claiming the animal fed on blood, or pursued anything, or did anything at all beyond drift. But a name outlives its context. The animal that came up in that net has been called the vampire squid ever since, in every language and every popular account, and for most of the 20th century, the name did the thinking for people. It suggested an appetite. It suggested a habit. It suggested that somewhere in the middle water there was a small, dark thing that lived by taking something from other animals. And that suggestion sat in the literature and in the public imagination for 100 years, unexamined, because there was no way to check it. The formal description was published in 1903. The full scientific results of the Valdivia expedition ran on for decades afterwards, volume after volume, and Chun's own detailed treatment of the cephalopods, with its beautiful plates, did not appear until 1910. He also wrote a popular account of the voyage for general readers, a book whose title translates as From the Depths of the World Ocean, and it did very well. People wanted to read about this. The deep sea in 1900 occupied roughly the position in the public mind that deep space occupies now, and Chun was writing dispatches from it. He died in 1914, in Leipzig, before most of the expedition's results were in print, and long before anybody worked out what his most famous animal was actually doing down there. There is something worth sitting with in that. The whole of the vampire squid's reputation was set in a single moment by a tired man in a converted cargo hold looking at a small, damaged animal in a tray, choosing 2 Latin words. Everything the animal has since turned out to be, its patience, its stillness, its almost complete disinterest in doing anything quickly, was invisible in that tray. It could not have been otherwise. You cannot see the life of a creature that lives by hanging motionless in the dark, by pulling it up through 800 meters of water in a net and looking at what arrives. Which brings us to the problem that shaped the next 50 years. Imagine, for a moment, that you wanted to study snowflakes, and the only tool you were permitted was a bucket. You could lower the bucket out of a window on a snowy night. You could haul it back up, and you would have, in the bottom of it, a small quantity of cold water and perhaps some slush. And from that you could establish, quite correctly, that snow is made of water and that it is cold. Everything else about a snowflake, its geometry, the way it turns as it falls, the fact that it is mostly air, the 6-fold symmetry that makes it what it is, would be gone. Not hidden, gone, destroyed by the act of collection. That is very close to what a plankton net does to a deep-sea cephalopod. The vampire squid is a soft animal. It has very little muscle for its size, and its tissues are loaded with ammonium salts held in fluid-filled spaces, which is a trick a number of midwater animals use to stay afloat without effort. Ammonium chloride solution is slightly less dense than seawater. Carry enough of it inside you and you weigh, in water, almost exactly nothing. The result is an animal with roughly the consistency of a firm jelly. An animal built to be held up by the water on every side of it. An animal that has never in its life had to support its own weight. Put that into a net and tow the net for an hour. The web tears. The arms bend backwards past the point where they would ever normally go. The filaments, which are extremely thin, snap off or retract so completely into their pockets that they are missed entirely. The skin, which is covered in small light-producing organs, abrades against the mesh and against the other animals in the cod end, and those organs come away. The pigment leaches. Then the whole thing comes up through 800 meters of water into pressure and temperature it has never experienced, and is put into a jar of alcohol, which fixes the tissue and pulls the water out of it and turns everything, in time, the same neutral tan. What sat in the trays of the world's museums for the first half of the 20th century, and what generations of taxonomists worked from, were shapes like that. Damaged, discolored, incomplete, and in many cases missing exactly the features that mattered most. So let us do it properly, with the benefit of a century of better tools, and describe the animal as it actually is when it is alive and undisturbed and hanging in its own water. Start with the size, because the popular imagination has never been reliable about this. An adult vampire squid has a body, the mantle, of perhaps 13 to 15 centimeters. With the arms extended and the web spread, the total length comes to something approaching 30 centimeters. In plain terms, it is about a foot long. You could hold one comfortably in 2 cupped hands, if it were the sort of animal that survived being held, which it is not. Every illustration you have ever seen that made it look like something the size of a car was working from the name rather than from the specimen. The mantle is rounded, slightly conical, tapering towards the rear. It is not the streamlined torpedo of a fast-swimming squid. It is a soft bell, and it is not really used for propulsion in the way a squid's mantle is. Along its sides sit a pair of fins, and these are the animal's engine. They are rounded and paddle-shaped, and they beat in a slow, flapping, almost unhurried way. And the effect, watched on video, is closer to a bird gliding than a fish swimming. Some observers have compared it to the pulsing of a jellyfish. Others, watching the fins move against the black, have said it looks like an animal flying very slowly in the dark, which is not a bad description at all. Inside the mantle, supporting those fins, there is a remnant of a shell. This matters more than it sounds. The whole history of the cephalopods is a history of losing the shell. The ancestors of every squid and octopus alive today lived inside coiled chambered shells like the nautilus still does. and over a very long stretch of time that shell was internalized, reduced, flattened, and finally, in the octopuses, almost entirely abandoned. Squid retain a stiff, transparent blade of it, the pen or gladius, running down the length of the mantle like a spine. Octopuses retain, at most, 2 small rods where the fins used to attach, or nothing at all. The vampire squid keeps a broad, thin, transparent gladius, leaf-shaped, wide enough to brace the fins against. It is an intermediate condition, and it is one of many features on this animal that sit awkwardly between the two great living groups. We will come back to that awkwardness, because it turns out to be the most interesting thing about the creature after what it eats. Now the arms. There are 8 of them, which is the octopus number, and they are joined to each other along most of their length by a continuous sheet of skin called the web, or the velum. When the arms are relaxed and spread, the web opens out into a shallow bowl with the mouth at the center of it. When the arms draw together, the web closes into a smooth, dark bell, and the animal becomes, briefly, a featureless shape hanging in the water with nothing to grip and nothing to see. Along the inner surface of each arm, where a squid would carry a dense double row of suckers all the way down, the vampire squid carries something more curious. There are suckers, but only along the outer half of each arm, and they lack the toothed horny rings that let a squid lock on to struggling prey. And interleaved among them, in a single row running the length of the arm, are the cirri. The cirri are soft, tapering, finger-like projections of tissue. They stand up from the arm like the teeth of a comb, or like the barbs on one side of a feather, and there are perhaps a couple of dozen on each arm. In old descriptions, they were sometimes called spines, which gave entirely the wrong impression, because a spine is hard, and these are not. They are pliable and sensitive, and, so far as anyone can tell, they are principally organs of touch and of handling. They are also, as we will see, a large part of how this animal eats. Between the first and second pair of arms, tucked into a small pocket on each side, are the 2 structures that gave the vampire squid its long-standing mystery. They are called the retractile filaments, and they look like nothing else in the cephalopods. Each one is a thin, pale thread, so fine that it is easily missed and easily broken, and it can be extended out of its pocket to a length considerably greater than the whole rest of the animal. When retracted, it coils away completely, and the animal appears to have only its 8 arms. When extended, it trails out into the dark water like a single long hair. Chun saw them. Everyone who examined a specimen carefully saw them. Nobody could say with any confidence what they were for, and the hypotheses put forward across the 20th century covered essentially every function a thread can have. It was proposed as a sensory antenna, and as an anchor line, and as a lure, and as a tether to something, and as a vestige of the feeding tentacles that squid use to strike at prey. That last idea was the most popular, and as it turned out, the most misleading, because it kept everyone's attention pointed at the question of what this animal was catching. Then the eyes. The eyes of a vampire squid are, in proportion to its body, among the largest of any animal that has ever been measured. In a fully grown individual, each eye is a sphere of roughly 2.5 centimeters across, set into a head that is only a few centimeters wide, which gives the animal the look of something drawn by a person with a fondness for very large eyes. They are globular, they bulge, and when a light is brought near them they can appear red, which is a reflection rather than a color. The reason for the size is straightforward. Where the vampire squid lives, there is essentially no sunlight. What light there is comes in flashes, from other animals, generated chemically, and it is almost always blue. To see anything at all in that setting, you need to gather every photon available, and the way you gather photons is with a large aperture. The eye of a vampire squid is a light bucket. It is tuned, as far as the pigments have been studied, to the blue-green wavelengths that carry furthest through seawater, and it appears to run on a single visual pigment, which means the animal almost certainly does not see color in any sense we would recognize. It sees brightness, and it sees it extremely well. The skin is where the last of the surprises are. Across the surface of the mantle, and the head and the outer face of the arms, scattered in their hundreds, are small light-producing organs called photophores. They are not evenly distributed, and they are not all the same. Most are tiny, little more than points. Behind the fins, on the upper surface of the mantle, sit 2 considerably larger organs, pale and rounded, which in some photographs look uncannily like a second pair of eyes. These larger organs can be covered. There is a fold of tissue that closes over them like an eyelid, so the animal can dim itself or extinguish itself at will. And the color of that skin, in life, is not black, though it is very often described as black, and photographs almost always render it that way. It ranges from a deep velvet red-brown through to something paler and more russet, and in some individuals to a shade closer to plum. The web between the arms tends darker than the outer skin. The reason the animal looks black in every image is that the images are taken under artificial white light. Down where it lives, the only light in the water is blue, and red pigment under blue light does not appear red. It absorbs. It reflects almost nothing back. An animal colored deep red, in water where only blue light exists, is not a red animal at all. It is a hole in the water. That is the body. About a foot of soft red-brown tissue, mostly water and ammonium, with a leaf of transparent shell inside it, 8 webbed arms lined with soft combs, 2 hidden threads, a pair of enormous light-gathering eyes, and a scattering of lamps across its skin that it can switch off. It is a strange assembly. It looks assembled from parts belonging to 2 different animals, and for a very long time that is precisely the problem it presented to anyone who tried to file it. Between Chun's description and the middle of the 20th century, the vampire squid was named again, and again, and again. Specimens kept arriving. A net off California brought up something small and dark with 1 pair of fins, and it was described and given a name. A net in the Atlantic brought up something rather larger with 2 pairs of fins, and that was clearly a different animal, so it was described and given a different name, in a different genus. Something from the Indian Ocean had a web that reached further down the arms. Something from the tropical Pacific had eyes set at a slightly different angle. Each of these went into the literature as a discovery, with a describer's name attached, and the group swelled. By the 1930s, there were roughly 11 named species distributed across 8 named genera, all of them founded on animals that had been dragged up through hundreds of meters of water in nets and preserved in jars, and all of them, as it turned out, the same creature. The thing that made the confusion almost inevitable is one of the most unusual features of the animal's life, and it concerns the fins. A very young vampire squid, newly hatched and only a few millimeters across, has a pair of fins set well forward on the mantle. It swims with them. In whatever slow way something a few millimeters long can be said to swim. As it grows, over months, a second pair of fins begins to develop behind the first. For a period, the animal has 4 fins, 2 pairs, which is a condition found in no other living cephalopod, and then the original front pair begins to shrink. It is reabsorbed. The tissue is taken back into the body, and the animal completes its growth with a single pair of fins in a different position from the pair it started with. Consider what that does to a museum drawer. A researcher opens 3 jars from 3 different oceans. In the first is a small animal with 1 pair of fins near the front. In the second is a medium-sized animal with 2 pairs. In the third is a larger animal with 1 pair of fins in the middle of the mantle. On the evidence in front of him, and with no way of watching a single individual grow, these are 3 animals. They differ in a character that in every other cephalopod group is stable and diagnostic. He describes 3 species and, being conservative about it, puts them in separate genera, because a difference in fin number is not the sort of thing you shrug at. Layer on top of that the fact that a young vampire squid is not simply a smaller version of an adult. The proportions change substantially with age. The web is relatively shorter in the young and grows to cover more of the arm. The eyes change shape as the animal grows, becoming more stalked in some intermediate stages and then settling back. The relative length of the arms shifts. The photophores develop progressively, and a young individual tends to be caught at a different depth from an old one. So even the ecological information seemed to point to distinct animals. The result was a small, tangled, entirely artificial family of deep-sea cephalopods, built out of 1 species and a great deal of honest confusion. The person who took it apart was Grace Pickford. She was born in England at the very beginning of the 20th century, in 1902, and she came from a family with a scholarly bent. She studied at Cambridge, went out to South Africa to work, and eventually came to the United States, where she took a doctorate in zoology at Yale in 1931, at a time when very few women were awarded doctorates in that subject at that institution. She spent much of her working life attached to the Bingham Oceanographic Collection at Yale, and she worked, for decades, in a research position rather than a professorial one, on a salary and with a title that did not reflect what she was producing. Late in her career, she became a full professor at Hunter College in New York. She also did substantial and quite separate work on fish endocrinology, which is a different field entirely, and she was good enough at it to write a standard reference. But her cephalopod work is what concerns us, and specifically her work on this one animal, which occupied her on and off for the better part of 20 years. She had an advantage that her predecessors did not, which was access to a great deal of material at once. The Danish research vessel Dana had spent the 1920s and early 1930s running expeditions across the world's oceans with fine nets, and the Dana collections held vampire squid specimens from many regions and, crucially, of many sizes. Pickford assembled those with material from museums in Europe and America, and instead of describing what was in front of her one jar at a time, she laid the whole series out and sorted it by size. And when you do that, the species dissolve. The small ones with front fins grade into the medium ones with fore fins, which grade into the large ones with middle fins. The web proportions form a smooth curve against body length. The arm ratios do the same. There is no gap anywhere in the series, no point at which one form stops and another begins, and there is no geographic pattern either, because animals from the Atlantic and the Pacific and the Indian Ocean fall along the same single line. What had looked like 11 species in 8 genera was one animal, growing up. Pickford published this across a set of monographs in the 1930s and 40s, the most substantial of them appearing in the mid to late 1940s, and the conclusion has stood for the better part of 80 years. One species, in one genus, in one family, and, as she and others recognized, in one order, containing nothing else that is alive. She did something else in that work that turned out to matter enormously later, and she did it almost in passing. She noticed where the specimens came from. The capture records included depth, and where they existed they included the temperature and the chemistry of the water at those depths. Pickford went through them and observed that the vampire squid was not simply a deep-sea animal in the general sense. It was concentrated, across every ocean where it had been found, in a particular and rather specific band of water. Most of her specimens came from between 600 and 900 meters, in water between about 2 and 6 degrees, and, more tellingly, in water where the dissolved oxygen was very low, not merely lower than the surface, low enough that the great majority of animals of comparable size and complexity simply cannot function there. She coined a word for the animal's apparent preference. She called it oligoaerobic, from the Greek for few and for air. An animal of little oxygen, it was an observation ahead of its evidence. Pickford could see the correlation in the capture records, but she could not explain how a cephalopod, a group with a well-earned reputation for high-energy living and high oxygen demand, was managing to hold a permanent address in a layer of water that other cephalopods pass through as quickly as they can. That explanation would take another 50 years and a different kind of science entirely, and when it came it would arrive from 2 directions at once, from physiologists measuring oxygen consumption in tanks, and from engineers building machines that could go down and watch. We are going to spend a long while in that layer of water tonight, because it is the whole key to the animal. But before we descend into it, there is a question left over from the century of confusion, and it is the one that was never really settled by sorting specimens by size. Pickford established what the vampire squid is not. It is not 11 species. It is not 8 genera. What it is remained, and to some extent remains, a genuinely difficult question, and it has to do with a family tree. The common name is wrong twice over, which is unusual even by the standards of common names. It is not a squid. It is also not an octopus, though it is much nearer to being one. And the reason it sits so uncomfortably between the two is that it split off from both of them a very long time ago and then simply carried on, alone, while everything around it changed. The soft-bodied cephalopods, the group that contains everything except the nautilus, divide into 2 great branches. On one side are the 10 armed forms, the squid and the cuttlefish and the bobtails, animals with 8 arms and 2 additional long feeding tentacles that can be shot out to seize prey. On the other side are the 8 armed forms, which is to say the octopuses and the vampire squid. That is the first correction. Whatever the name says, this animal belongs on the octopus side of the tree. It has 8 arms and no feeding tentacles. Its nearest living relatives, as far as anatomy, gene sequences, and every reanalysis of the past 2 decades can establish, are the octopuses, and in particular the deep-sea octopuses with fins and webs and combs along their arms, the ones called cerates. But it is not one of them either, and the distance is not small. The vampire squid occupies its own order, Vampyromorphidae, which is a rank in the classification roughly equivalent to the rank that contains all the octopuses together, or all the true squid together, and that order contains, alive today, precisely one thing. There is a particular quality to a taxonomic group of one, It is a category that exists only because everything else that would have been in it is gone. Somewhere in the deep past, there were other members, relatives, cousins, an entire branch with its own variety and its own experiments, and what remains of that branch is a single soft red animal about a foot long, hanging in the middle water of the warm oceans, still doing whatever its lineage has been doing. That is where the phrase living fossil gets used, and it is worth being careful with it, because it carries an implication that is not true. A living fossil is usually understood to mean an animal that has stopped evolving, a survivor from an earlier world that has been passed over by time. No such animal exists. The vampire squid has been subject to exactly as much evolution as every lineage alongside it, for exactly as long. Its ancestors were not sitting still while the fish and the octopuses got on with things. Every generation of them was tested and sorted like every other generation of every other creature on the planet. And the animal we have now is the current result of that process, not a leftover from an old one. What is true is that its lineage is ancient, that it retains a combination of features which in other groups have been lost or gone further, and that the branch has narrowed almost to nothing. That is a story about extinction, not about stasis. We know quite a lot about what the branch used to hold, because vampyromorphs fossilize, occasionally, in the right conditions. Soft-bodied animals hardly ever leave a record. The chances of a creature with no bones and no shell being buried fast enough, in water still enough and chemistry strange enough, to leave any trace at all are extremely poor. But there are a handful of places on Earth where those conditions were met, and where entire communities of soft animals were preserved in detail that seems almost unreasonable. One of them is a hillside in southeastern France, near a village called La Volte-sur-Rhône. The rocks there formed on a Jurassic seafloor roughly 160 million years ago, in what appears to have been a fairly deep basin, and they contain soft-bodied cephalopods preserved in 3 dimensions in mineral nodules, with arms, suckers, muscle fibers, and internal organs still discernible. Among them is an animal called Vampyroteuthis rodanica. Vampyroteuthis is a vampyromorph. It has the general architecture, the 8 arms, the web, the fins. For a long time, it was assumed to have lived much as the modern vampire squid lives, which would have made the animal's quiet gleaning habit an ancient one, an inheritance from the Jurassic. Then, a few years ago, researchers put the fossils through a synchrotron and reconstructed the internal detail of the arms and the sucker arrangement, and the picture that came back was of an animal noticeably better equipped for taking hold of living things than its modern relative. The arrangement of suckers and cirri along the arms suggested grasping and manipulation rather than gathering. Vampyroteuthis, on that reading, was an active predator. And then there is a fossil from Luxembourg, described in 2022, from rocks roughly 180 million years old. It is a vampyromorph, and it was buried holding 2 small fish in its arms. Not near them, holding them. The animal died in the middle of a meal, and the meal is still there in the stone. Put those together, and the shape of the story changes. The vampire squid's ancestors hunted. They caught fish and held them and ate them, in the ordinary way that soft-bodied cephalopods have always made a living. The stillness, the patience, the extended filament, and the drifting collection of what falls, all of that is not ancient at all. It is a departure. Somewhere along the line, in a branch of hunters, one lineage stopped hunting. That is a more interesting fact than the living fossil version, and it changes what you are looking at when you watch the animal hang motionless in the dark. You are not watching something that never learned to chase. You are watching the descendant of chasers that gave it up and did so successfully enough that it is still here after every one of its more energetic relatives has gone. Why they gave it up is a question we cannot answer directly, but we can see the conditions under which giving it up would pay. Hunting in the deep sea is expensive, and the returns are uncertain. Prey is sparse. Water is dense. Every meter you move costs something, and every meter you move you might not find anything. Meanwhile, in that same water, there is a continuous, reliable, entirely unresisting supply of organic material falling through from above, requiring no pursuit, no strike, no struggle, and no risk. It is poor food. It is low in energy, and much of it is already partly digested by something else. But it never stops arriving, and nothing has to be caught. An animal that can lower its energy requirements far enough to live on that supply gains access to something rare, which is a habitat with almost no competition. Nothing else with a cephalopod's body plan can afford to be there. And there is a further advantage, which is the one that ties this whole story together. And it has to do with the particular layer of water where the falling snow passes through a band of very low oxygen. That is where we are going now. Let us go down. Start at the surface on a calm day in the tropical Pacific, a long way from anywhere, where the water is the deep blue that only happens where there is nothing suspended in it. The top few meters are warm. Sunlight comes down through them in moving columns, and the whole layer is stitched with light. There is more dissolved oxygen in this water than anywhere else in the ocean, and it comes from two places. Some of it is simply the atmosphere, pressed into the surface by wind and wave and left there. The rest is made in place by the countless small photosynthetic cells that drift here and split water and release oxygen as a byproduct, exactly as a leaf does. This is the layer everything else depends on. It is thin. In clear ocean water, the light useful for photosynthesis is largely gone by 100 meters and effectively finished by 200, so the entire productive skin of the sea is thinner than the height of many buildings, spread over 2/3 of the planet. Sink through it. At 50 meters, the reds have gone from the light. Water absorbs the long wavelengths first, so red disappears almost immediately, then orange, then yellow. And by the time you are at any real depth, the world is rendered entirely in blue. At 100 meters, it is a dim blue afternoon. At 200, it is the blue of very late dusk. At 300, there is still light, but there is no longer enough of it to see anything by. Only enough for a well-adapted eye to detect the difference between above and below. This is the twilight zone, the mesopelagic, and it runs from around 200 meters to around 1,000. The temperature has been dropping the whole way. You pass through the thermocline somewhere in the first 200 or 300 meters, a boundary layer where the warm surface water sits on the cold, deep water and mixes with it reluctantly, and below that, the temperature falls into the single figures and keeps going. At 600 meters, in most of the warm oceans, you are in water of perhaps 4 or 5 degrees. The pressure is 60 times what it is at the surface. It is completely dark to a human eye, and somewhere in here, the oxygen begins to run out. It is worth understanding why, because it is not obvious. The ocean is made of water, and water is made partly of oxygen. But that oxygen is locked into the molecule, and no animal can breathe it. What a gill extracts is dissolved oxygen gas, the free molecules of it that are mixed into the water, and there is not very much even at the best of times. A liter of well-aerated surface seawater holds only a small fraction of the oxygen that a liter of air holds. Marine animals have always lived on thin rations by comparison with anything that breathes air. That dissolved oxygen enters the sea in only 2 ways, both of them at the surface. It comes from the atmosphere and it comes from photosynthesis, and both of those processes happen in the top 100 meters or so and nowhere else. Below that, there is no source, not one. Every molecule of oxygen in the deep ocean got there by being carried down. There are 2 ways it gets carried. The slow, global way is that at high latitudes, in the far North Atlantic and around Antarctica, surface water becomes cold and salty enough to sink, and when it sinks, it takes its dissolved oxygen with it and spreads out along the bottom of the world, filling the deep basins over a period of centuries. That is why the deepest water in the ocean, even at 4,000 or 5,000 meters, is reasonably well supplied. It was at the surface not so very long ago, in geological terms, and it has not been used up yet. The faster and more local way is simply mixing. Wind and turbulence stirring oxygen down from the surface into the layers immediately beneath. Now put against that the consumption. Everything that is made in the sunlit surface eventually dies and sinks, and as it sinks, it is eaten, not usually by large animals, mostly by bacteria, which colonize the sinking particles and break them down. And that breaking down is respiration, and respiration consumes oxygen. Every fragment of organic material falling through the water column is, in effect, a small, slow fire, and the fuel is the dead surface, and the air is the dissolved oxygen in the water around it. So there is a band of depth below the sunlight where no oxygen is being made, and above the deep water that arrived recently from the poles, where oxygen is only ever being spent and never replaced. The sinking rain of organic matter passes through it and is consumed in it, and in the parts of the ocean where the surface is most productive and where the circulation is most sluggish. That band can be drawn down remarkably far. These are the oxygen minimum zones. They are found beneath the most fertile surface waters in the world, off the coast of Peru and Chile, where upwelling brings nutrients up and the surface blooms with life. Off Namibia, for the same reason, in the Arabian Sea and the Bay of Bengal, in the eastern tropical Pacific, over an area larger than several countries put together, and, importantly for our story, off the coast of California and in the Gulf of California, where a good deal of the research that finally explained the vampire squid was done. There is a detail in that list which explains the whole geography of our animal, and it is easy to miss. Every one of those places is warm or temperate. There is no strong oxygen minimum zone in the Arctic. There is none around Antarctica. There is none in the far North Atlantic. And the reason is that those are precisely the regions where the deep water is made. Cold surface water is denser than warm surface water, and salty water is denser than fresh. In the far North Atlantic and around the edges of Antarctica, the surface gets cold enough and salty enough that it becomes heavier than the water beneath it, and it sinks in great slow columns, carrying a full load of dissolved oxygen down with it. That water then spreads out along the bottom of the world and creeps away towards the equator over a period of centuries. So at high latitudes, the deep water has been at the surface recently and is well supplied. There is no room for an oxygen minimum to develop, because the layer is being refreshed from above faster than the sinking snow can burn it out. Move towards the tropics and the situation reverses. The surface is warm and it stays warm and it never sinks. The water at middle depths there has not touched the atmosphere for a very long time, and it arrived from somewhere else, already partly depleted, and it has been sitting there being consumed ever since. Overlay that with the intense biological production of an upwelling coast or an equatorial current system, and the oxygen at 600 meters has nowhere to come from and everything to be spent on. This is why you will not find a vampire squid off Iceland or in the Southern Ocean. Its distribution maps onto the warm and temperate oceans of the world, and within those, onto the layers where oxygen is at its lowest, because that is not merely where it can live, it is where its whole strategy makes sense. In well-oxygenated water, the animal would have no refuge, because everything that currently cannot follow it there would be able to. The barrier that keeps others out is the same barrier that keeps it safe. In the core of a strong oxygen minimum zone, the dissolved oxygen can fall to a few % of what the surface holds. Measurements from the water in which vampire squid have been taken include values as low as around 3% of saturation. For most large active animals, that is not a difficult environment. It is an impossible one. The reason is arithmetic. An animal's muscles need a supply of oxygen delivered by blood. The blood picks it up at the gills, and the amount it can pick up depends on how much is in the water and on how strongly the animal's respiratory pigment binds it. Bring the water concentration low enough, and the gradient across the gill collapses. There is nothing to extract. A fast-swimming squid entering that layer is in the position of a runner at very high altitude, except that the altitude is worse than any mountain, and there is nothing to be gained by acclimatizing because the animal will run out of oxygen before it adapts. So most things do not stay. What happens instead is one of the great daily events of the planet. Enormous numbers of small animals, lanternfish and shrimp and copepods and squid, spend the daylight hours down in or just above the low oxygen layer, where the light is too dim for visual predators to work efficiently, and then rise at dusk, hundreds of meters, up into the productive surface to feed under cover of night, and sink back down again before dawn. It is the largest migration of animal biomass on Earth, and it happens twice every 24 hours, everywhere, and almost nobody watches it. Those animals are visitors. They tolerate the low oxygen for a shift and then they leave. Many of them are running on strategies that amount to holding their breath, dropping their metabolism, going still and dark and waiting out the day, and then paying the debt back later in oxygen-rich water. The vampire squid does not leave. That is the single most remarkable fact about it, and it is the fact around which everything else about its body is arranged. It lives at 600 to 900 meters, sometimes shallower, sometimes as deep as 1500, and it lives there permanently. Not for the daylight hours, not while waiting for something. It appears to complete its entire life cycle, from egg through juvenile to adult and through however many spawnings it manages, inside a band of water that other cephalopods enter only briefly and leave as soon as they can. It is the only cephalopod known to do this. And when Pickford noticed that pattern in the capture records of the 1940s and coined her word for it, she had identified the animal's central adaptation without yet having any way to see how it worked. The how turns out to be several things at once, layered together, none of them individually sufficient. It is in the blood, it is in the gills, it is in the muscle, or rather in the near absence of it. It is in the way the animal holds itself up, and in how much it moves, and in what it does with its time, and it is above all in the extraordinary lowness of its demand. The blood of a vampire squid is blue, not metaphorically. All the cephalopods, and the crustaceans, and a good many other invertebrates, carry oxygen using a protein called hemocyanin. And where our own hemoglobin is built around iron, hemocyanin is built around copper. Iron bound to oxygen is red. Copper bound to oxygen is blue, a pale washed-out blue like weak ink, and cephalopod blood carries that color when it is oxygenated and turns nearly clear when it is not. There is another difference that matters more than the color. Our hemoglobin is packed into red cells, billions of them, which lets us carry a very large amount of the protein in a small volume of blood without making the blood impossibly thick. Cephalopod hemocyanin is not in cells. It floats free, dissolved in the plasma, in very large molecules, and that puts a ceiling on how much of it there can be. Cephalopod blood is, in general, a less efficient oxygen carrier than ours by a considerable margin. They compensate by pumping it hard and fast, with 3 hearts, 2 of which serve only the gills. So a cephalopod already begins from a difficult position, and then this one went and moved into the least oxygenated water in the open ocean. What it has done with its hemocyanin is the first part of the answer. Every oxygen-carrying protein has a property called affinity, which describes how tightly it holds on to oxygen at a given concentration. High affinity means the protein will pick oxygen up even when there is very little of it about. Low affinity means it needs a rich supply to load it all, but releases what it has readily. There is a trade-off built into that, and it is unavoidable. A protein that grips oxygen tightly enough to strip it from very poor water will also be reluctant to let go of it in the tissues where it is needed. You cannot have both. You can only choose which end of the problem to optimize for. When researchers measured the oxygen-binding properties of vampire squid blood, they found an affinity higher than any other cephalopod that had been tested, and higher than most of the well-known examples of low-oxygen specialists among the mollusks generally. The blood of this animal will load with oxygen at concentrations where the blood of an octopus or a market squid would remain essentially empty. It is tuned right to the extreme of one end of the trade-off, which tells you something immediately about the other end. A blood that grudging about releasing oxygen can only work in a body that is not asking for much. The second part of the answer is the gills. They are large, with a great deal of surface area for the size of the animal, which is what you would design if you needed to extract as much as possible from water that has very little to give. Surface area is the currency of gas exchange, and this animal has invested in it. The third part, and by some distance the largest, is that the vampire squid has one of the lowest energy demands ever measured in an active animal of its kind. In the 1990s, a group of physiologists working off California and Hawaii undertook one of those studies that quietly rearranges a field. They caught 33 species of open ocean cephalopods, brought them up carefully, put them into sealed respirometry chambers at the temperatures they normally live at, and measured how fast each one consumed oxygen. Then they plotted those rates against the minimum depth at which each species occurred. The relationship was remarkably clean. The shallower and more active the animal, the higher the rate. A vertically migrating squid of the genus Gentlest, a muscular, fast, visually hunting animal, sat at the top of the range. And at the very bottom, by a wide margin, was Vampyroteuthis. The difference between those two ends was on the order of 100-fold. One individual vampire squid recorded the lowest mass-specific rate of oxygen consumption that has ever been measured for any cephalopod. In practical terms, it means that the amount of energy this animal spends simply existing, per gram of itself, per hour, is closer to that of a sea anemone than to that of anything else in its own class. Some of that is temperature. Cold slows chemistry, and an animal living at 4 degrees will always burn less than the same animal at 20. But the researchers were careful about this, and the vampire squid's rate is low even after temperature is taken into account. Something else is going on, and the explanation that emerged has an elegance to it. In the bright surface ocean, animals find each other by sight, at range. A predator can see prey from many meters away, and prey can see the predator. And once that is true, the whole encounter becomes a race. Selection favors speed, acceleration, endurance, and the muscle and the metabolic machinery to support all three. The whole arms race is driven by long sight lines. Take away the light, and the sight lines collapse. In the deep middle water, nothing sees anything until it is very close. Encounters are brief and local. There is no advantage in being able to outrun something you will never see coming, and no advantage in being able to run down something you will only detect at arm's length. And the moment speed stops paying, the enormous ongoing cost of maintaining the equipment for speed becomes pure waste. So, metabolic rates fall with depth across the whole visual animal kingdom, in fish and crustaceans and cephalopods alike, and they fall further than temperature alone can explain. The deep sea is full of animals that have quietly stopped paying for an ability they no longer use. The vampire squid has gone furthest of any cephalopod down that road, and you can see it in the body. There is very little muscle. What there is thin and pale and poorly supplied with the internal machinery that muscle uses to burn fuel aerobically. The mantle, which in a squid is a thick barrel of muscle capable of contracting hard enough to fire the animal backwards, is in the vampire squid a soft bag. It can jet. It does jet occasionally, when startled, and the results are unimpressive and short-lived. Observers who have watched it try have noted that a few contractions exhaust it, after which it needs a long time to recover, sometimes hanging still for many minutes afterwards. Escape by speed is simply not a strategy available to it. Instead, there is water. The tissue of a vampire squid is loaded with fluid-filled spaces containing a solution rich in ammonium ions. An ammonium chloride solution is very slightly less dense than seawater. Carry a large enough volume of it, and you cancel out the weight of everything else. The shell remnant, the beak, the tissue, the organs. The result is an animal with almost exactly neutral buoyancy, which means it neither sinks nor rises and does not have to do anything at all to stay where it is. Consider what that actually buys. A fish without a swim bladder must swim continuously or sink. A shark must keep moving. A squid must either jet or beat its fins. All of them are paying a permanent tax simply for the privilege of remaining at a given depth. The vampire squid pays nothing. It can hang, motionless, at 800 meters, indefinitely, spending no more energy than it would spend lying on a shelf. And to make use of that, it has a superb sense of which way is up. The statocist, the balance organ set into the cartilage of the head, is unusually well developed in this animal, larger and more elaborate than a creature of its size and sluggishness would seem to require. It contains dense mineral grains resting on sensory hairs, and it reports, continuously and precisely, the animal's orientation and any acceleration acting on it. An animal that spends its life suspended in a fluid with no floor, no ceiling, no horizon, and almost no light needs to know its attitude at all times, and this one does. Add it together, and you have a coherent design. A body that weighs nothing in water, so it need not move. Blood tuned to extract oxygen from water that has almost none, at the cost of releasing it reluctantly, which is affordable because almost nothing is demanding it. Gills built to maximize contact with that water. Muscle reduced to the minimum that will still allow slow flapping and occasional retreat. A metabolism running so low that the animal's food, poor as it is, lasts a very long time inside it. And an excellent sense of balance, because in that environment, orientation is one of the few things worth knowing. It is a design entirely built around not doing very much, and it is only viable if the animal can eat without hunting. Which brings us, at last, to the snow. In the summer of 1934, off the coast of Bermuda, a naturalist named William Beebe was lowered into the Atlantic inside a steel sphere 4.5 feet across, with 2 small quartz windows and a telephone cable running back up to the ship. The sphere was called the bathysphere. It had no propulsion and no way of returning to the surface under its own power, and the men inside it were entirely dependent on a winch operator on a rolling deck. Beebe went down, on that expedition and others, further than anybody had been, and he spent his time at depth doing what he had always done, which was to look out of the window and write down what he saw. Among the things he described, and one of the descriptions that has lasted longest, was a continuous fall of small pale particles through the beam of the sphere's searchlight. It looked, he said, like snow. The phrase stuck. Marine snow is now the standard term for it, in textbooks and papers and expedition logs, and it is one of those rare pieces of scientific vocabulary that is both technically precise and genuinely beautiful. What it is the ocean's falling remains. Consider what the sunlit surface of the sea actually does. It grows. Every hour of daylight, across the whole illuminated skin of the planet's water, single-celled algae are dividing, building tissue out of dissolved carbon and light. Diatoms in their glass boxes, coccolithophores plated in chalk, dinoflagellates and cyanobacteria, and countless things too small to have common names. That growth feeds the copepods, which are grazing shrimp-like animals a millimeter or two long, present in numbers that are essentially uncountable, and the copepods feed everything else. And all of it, without exception, ends. Cells die and their contents leak away and their casings sink. Copepods die. Copepods also, while alive, produce fecal pellets, small dense packets of processed algae bound in a membrane, and they produce them constantly, and those pellets sink faster than anything else in the system because they are compact and heavy. Jellyfish and salps and comb jellies die and come apart into transparent shreds. Fish scales fall. Molted shells fall. Bacteria colonize everything and knit it together with sticky secretions, and the small pieces clump into larger pieces, and the larger pieces become soft, ragged flocks that drift down like ash. There is one contributor that deserves a moment on its own, because it is strange even by the standards of this subject. Among the animals of the open water are the larvaceans, small tadpole-shaped creatures related, distantly and surprisingly, to ourselves. A larvation feeds by building a house. It secretes, out of mucus, an elaborate transparent structure around itself, with intake filters of extraordinary fineness, and it sits inside beating its tail to draw water through the filters and strain out particles far too small for anything else to catch. The house works beautifully, and it clogs quickly. When it clogs, the animal abandons it, swims out, and builds another, and it does this several times a day, every day, for its whole short life. The abandoned houses sink. They are large, they are laden with everything the filters caught, and there are unimaginable numbers of them. In some parts of the ocean, discarded larvation houses are among the single largest contributors to the material falling into the deep. An entire architecture of clear mucus, built and abandoned and built again, drifting down through the dark in its millions. All of this together is the snow. It does not fall the way rain falls. Most of it is very nearly the same density as the water it is falling through, which means it descends slowly and it wanders. Small particles may take weeks to travel a few hundred meters. Larger aggregates, or dense fecal pellets, can manage 100 meters in a day or more. Individual flakes turn as they go, catching whatever faint light there is, spiraling in the microscopic eddies of an ocean that is never entirely still. Watched from a submersible, the effect is genuinely of a snowfall, except that it never lands within sight and never stops and has been going on continuously for something like the last 500 million years. As it falls, it is eaten. Bacteria are on every particle within hours, breaking the material down and consuming oxygen as they do so, which is the process that carves out the oxygen minimum in the first place. Small crustaceans intercept flakes and eat them. Filter feeders strain them, and a great deal of what is eaten is excreted again as new particles that continue downward. The snow is repackaged over and over on its way down, and each repackaging strips more of the energy out of it. The arithmetic of that is stark. Of all the organic material produced by photosynthesis at the surface of the open ocean, only a modest fraction ever begins to sink at all. Of that fraction, the great majority is consumed in the upper few hundred meters. By 1,000 meters, only a small percentage of the original surface production is still falling, and by the abyssal floor it is a percentage of a percentage. What arrives at the bottom is the residue of a residue, and it settles there as a fine, pale ooze that has been accumulating for so long that in places it is kilometers thick. Scientists call the whole process the biological pump because it moves carbon from the atmosphere into the deep ocean and stores it there, and it is one of the reasons the planet's climate is what it is. But at 600 meters, where our animal lives, the snow is still very much falling, and there is a great deal of it. That is the crucial geographic coincidence at the center of this story, and it is worth stating plainly. The strong oxygen minimum zones exist precisely because the surface above them is exceptionally productive. Heavy production means heavy sinking. Heavy sinking means heavy bacterial respiration at depth, and heavy respiration is what strips the oxygen out. The two things are the same phenomenon seen from two sides, which means that the layer of water where almost nothing can breathe is also, necessarily, the layer with the most food falling through it. That is an extraordinary situation. There is a band in the ocean, hundreds of meters thick, spanning entire ocean basins, in which a continuous supply of edible material is drifting past, unclaimed, because almost nothing can afford to stay there and collect it. It is not a barren place. It is a well-stocked place with the doors barred. The vampire squid found the way in. It did so, as far as we can reconstruct, by lowering its energy demand until it fell below the ceiling that the oxygen imposes, which meant giving up muscle, giving up speed, giving up hunting, and accepting a diet that almost no other cephalopod could survive on. Everything we walked through in the last section, the blood, the gills, the ammonium, the abandoned musculature, is the price of admission. And in return, it gets a habitat with abundant food, very few competitors, and very few predators. And it gets to spend its entire life there without ever needing to leave, none of which was known for certain until quite recently. For most of the 20th century, the vampire squid was assumed to be a predator of some sort, because its name implied it and because that is what cephalopods do. The stomach contents of netted specimens were examined several times over the decades, and the results were inconclusive, largely because the stomachs of animals hauled up in nets are full of whatever else was in the net. To settle it, somebody was going to have to go down and watch. There is a canyon in Monterey Bay, on the coast of California, and it is the reason a great deal of what we know about the deep middle water is known at all. It begins astonishingly close to shore, off the town of Moss Landing, a few hundred meters from a beach where people walk their dogs. The seafloor drops away into a submarine canyon comparable in scale to the Grand Canyon, and it runs out and down into water thousands of meters deep. In most parts of the world, reaching the deep ocean means a voyage of days. In Monterey Bay, you can be over 1,000 meters of water within an hour of leaving the dock, and back at the dock the same evening. In 1987, David Packard, who had made a fortune in electronics, put a large amount of money into founding a research institute on that shoreline, with a specific and somewhat unfashionable idea behind it. The idea was that ocean science had been limited for a century by its instruments, and that the way to advance it was to build better machines and then hand them to biologists. The institute built remotely operated vehicles, not submarines with people inside, which are expensive and dangerous and limited by how long a human can sit in a cold sphere, but tethered robots on a fiber optic cable, carrying lights and high-definition cameras and manipulator arms and a set of sampling containers. A vehicle like that can stay down for a full working day. It can hover. It can follow a single animal at a fixed distance and watch it without touching it, for as long as the ship above can hold station. Among the scientists who took hold of that capability was a midwater ecologist named Bruce Robison, and he has spent a working lifetime doing something that sounds simple and is not, which is watching animals in the open water of the deep sea behave normally. Everything before that had been done on corpses. Everything. A century of deep-sea biology built on the contents of nets. What the vehicles offered was the chance to see the living animal in its own water, undisturbed, doing whatever it does when nobody is interfering, and the results across many species have repeatedly overturned what the specimens implied. Monterey Bay has, off the shelf and down the canyon, a well-developed oxygen minimum zone, and in that layer, at depths between roughly 600 and 900 meters, there are vampire squid, not many but reliably, year after year. Over 2 decades, the Institute's vehicles accumulated a very large archive of video, cataloged frame by frame, and within that archive were a substantial number of sequences of Vampyroteuthis simply going about its life. In 2011 and 2012, Robison and a Dutch cephalopod biologist named Hendrik Hoving went through all of it. They did not only watch video. The study they eventually published was assembled from 4 separate lines of evidence, and it is worth listing them, because it is the combination that made the conclusion stick. They analyzed the archived in situ footage of vampire squid in the wild, frame by frame, looking at posture and behavior. They brought live animals up carefully in sealed containers, kept them in cold, dark tanks, and offered them things, watching what they did with each. They examined the stomach and gut contents of preserved specimens from museum collections around the world, identifying the fragments in them, and they put the filaments, the arm suckers, and the cirri under optical and scanning electron microscopes to see what these structures are actually built to do. The picture that came out of it was consistent from all 4 directions, and it was not the picture anyone had been expecting. Start with the video, because the video is the part that would have told you if anyone had looked hard enough sooner. A vampire squid at depth, undisturbed, is very often found in a particular attitude. It is oriented more or less horizontally in the water, motionless, with the mantle level and the arms and web relaxed. And from one side of the arm crown, a single filament is extended, out and away, straight into the water, held there, only one at a time. The other stays coiled in its pocket. The extended one is very much longer than the animal, a fine pale line running off into the dark beyond the reach of the lights, and the animal does not move it, does not sweep it, does not wave it about. It simply holds it out and waits. The vehicles recorded animals holding this position for extended periods with no other activity at all. Under the microscope, the filament turned out to be densely covered in fine sensory structures along its length. It is not a hook, and it is not sticky in the way a spider's line is sticky. It is a sensor. It is 100 centimeters of nerve laid out into the falling snow. And what it does is register contact. When something touches it, the animal responds. The sequence recorded both in the wild and in the tanks runs like this. The filament is drawn back in towards the arm crown. As it comes in, it is pulled through the web, between the arms, past the suckers. The suckers on the arms of a vampire squid, unlike those of a squid or an octopus, are not primarily gripping organs, and what they do instead is secrete mucus. The particles adhering to the filament are stripped off and bound up in that mucus into a single soft mass, a bolus, and then the cirri, those soft comb-like fingers running along the inside of each arm, work the bolus down the arm and pass it in towards the mouth. The whole operation is slow, deliberate, and entirely without struggle. Nothing is caught, nothing resists. The animal is, in the most literal sense available, gathering, and the gut contents confirmed exactly what you would predict from that. What Hoving and Robison found inside vampire squid stomachs was detritus in a range of sizes, from small particles up to sizable aggregates. The identifiable components included the remains of gelatinous zooplankton, discarded larvation houses, fragments of crustacean shell and body, the glassy frustules of diatoms, and fecal pellets. There were no intact prey animals. There was no pattern of freshly captured, freshly killed anything. It was marine snow. All of it, or so nearly all of it, as makes no difference. It is worth following the bolus the rest of the way in, because the equipment at the end of the arms tells the same story as everything else. At the center of the web, where the 8 arms meet, is the mouth, and every cephalopod's mouth is built around a beak. It is made of hardened chitin, the same material as an insect's shell, stiffened at the tip and flexible at the base, in 2 parts that close past each other like a parrot's bill, with the lower half riding outside the upper. In a hunting squid or a large octopus, that beak is a serious instrument, capable of taking pieces out of a fish or cracking the shell of a crab, and it is worked by a substantial block of muscle. The beak of a vampire squid is small, pale, and relatively delicate. It is not the beak of an animal that expects resistance. It is closer to a pair of soft forceps than a cutter, and it handles a mass of mucus-bound detritus without difficulty, because that is the only thing it is ever asked to handle. Behind it sits the radula, which is a structure found across the mollusks and which has no equivalent in our own anatomy. It is a ribbon carrying rows of small backward-pointing teeth mounted over a supporting cushion, and it works rather like a conveyor, drawing material inward. In a limpet, the radula scrapes algae off rock and is hard enough to wear grooves in stone. In a hunting cephalopod, it rasps flesh. In the vampire squid, it simply moves a soft parcel along. Everything about the front end of this animal, in other words, is scaled for material that does not fight back, and that scaling is consistent with the video, with the microscopy, and with the gut contents. 4 independent lines of evidence, and none of them describe a predator. There is one more consequence of the finding, and it is broader than the animal itself. Before 2012, the middle water of the ocean was generally described as a predator's world, a place where sparse animals hunt each other in the dark. That description was built almost entirely on stomach contents, which is to say on the animals that eat other animals conspicuously enough to leave identifiable remains. Detritus does not leave identifiable remains. It leaves a stomach full of brown mush, which a century of researchers looked at, could make nothing of, and set aside. The vampire squid was the case that showed how much that method might be missing. When the cameras went down and stayed down, an animal that everybody had confidently filed as a predator turned out to be the ocean's most patient collector of falling waste. And the question that follows is obvious. How many others are there? How much of the Middle Ocean's economy runs not on hunting, but on the quiet interception of what drifts down, in ways that no net and no dissection would ever have revealed? Nobody yet knows. But the answer is almost certainly more than anyone assumed in 1990. The vampire squid is a detritivore. It is the only cephalopod known to be one, in a class of roughly 800 species, otherwise composed entirely of predators, and it makes its living by holding a thread out into the dark and collecting what the surface of the ocean has finished with. There is a further point in the microscopy that closed an old question at the same time. The filaments had been argued over for a century, with the most popular hypothesis holding that they were the remains of the two feeding tentacles that squid used to strike. The detailed anatomy did not support that. The filaments are, in their structure and their innervation and their position, modified arms. They correspond to a pair of arms, not to squid tentacles, and their transformation into thin sensory threads is a development unique to this lineage. So the animal has 8 arms, like an octopus, and it has taken one pair of what would have been arms and turned it into a pair of fishing lines for dust. There was one more piece of confirmation, and it came later and from an entirely different direction. Every animal carries, in its own tissue, a chemical signature of what it has eaten. The ratios of the heavy and light isotopes of nitrogen shift in a predictable way each time material passes from one organism into another. So an animal that eats a predator that ate a herbivore carries a higher nitrogen signature than the herbivore does. Measure the isotopes and you can place a creature on a scale of how far up the food chain it is feeding, without ever seeing it eat. In 2019, an international group did that for vampire squid, using specimens from many parts of the world, and compared them against a large set of other cephalopods. The vampire squid came out where the feeding studies said it should, at a distinctly lower position than the predatory cephalopods, and with a signature unlike anything else in the class. Independent method, same answer. It is worth pausing on how thoroughly this turns the animal's reputation inside out. Chun named it for blood. For 100 years it carried that name, and the name did the work of a description, and the deep sea being what it is, nobody could check. Museum drawers filled with damaged specimens. Popular articles were written, illustrations were drawn with the arms flared and the cirri rendered as fangs, and the whole apparatus of a small dark predator was constructed around an animal nobody had ever watched. And when the machines finally got down there and the lights came on and somebody spent 20 years looking, what they found was a soft creature about a foot long, weighing nothing, hanging perfectly still in the cold, with a single thread held out into the falling remains of the sunlit world, waiting for something to drift into it. One caveat, stated honestly, because it belongs here. The finding is that the vampire squid feeds principally and characteristically on detritus, and that its anatomy and behavior are built around doing so. It is not proven that it never takes a living thing. A small animal that blunders into the filament and is drawn in with everything else is not going to be distinguishable from a piece of marine snow in a stomach sample, and it would be surprising if that never happened. What is established is the mode. This animal does not pursue, does not strike, and does not need to. Every cephalopod you have ever heard of has an ink sac. It is one of the defining possessions of the group, and it is old. A gland attached to the gut produces a concentrated suspension of melanin, the same pigment that colors hair and skin, and stores it in a reservoir that opens into the funnel. When the animal is alarmed, it releases a quantity of that suspension into the outgoing jet of water, and a dark cloud appears in the sea where the animal was. The mechanism is cleverer than it first appears. The ink is mixed with mucus, and depending on how the animal releases it, the cloud can behave in 2 quite different ways. Released loosely, it disperses into a broad smokescreen, and the cephalopod changes color and direction and leaves through the far side of it. Released tightly, with more mucus and less water, it holds together into a compact blob roughly the size and shape of the animal that made it, hanging in the water and slowly drifting. Biologists call that a pseudomorph, a false body, and there is good evidence that a striking predator will commit to it while the real animal, now pale and moving, goes elsewhere. There is chemistry in it too. Cephalopod ink appears to interfere with the chemical senses of some predators, so that even an animal hunting by smell finds the trail suddenly confused. It is an excellent system, refined over a very long time, and it depends entirely on one thing. Somebody has to be able to see the cloud. At 800 meters, in the core of an oxygen minimum zone, nobody can. There is no ambient light. A cloud of black pigment released into black water is not a smokescreen and not a decoy. It is nothing at all. Whatever advantage ink confers in the upper ocean simply evaporates in the dark, and carrying the gland and filling the reservoir costs energy that this particular animal cannot spare. So the vampire squid has no ink sac. It is one of very few cephalopods that has lost it entirely, and it has replaced it with the deep sea solution, which is to do the same trick in reverse. In 2003, Robison and colleagues at Monterey published a study of light production by the arm tips of Vampyroteuthis, working from live animals brought up and held in cold, dark tanks and then gently disturbed while cameras watched. What the animal does, when something contacts it in a way it does not like, is eject a cloud of glowing mucus from the tips of its arms. The mucus is sticky and it is loaded with thousands of discrete luminous particles, small glowing spheres suspended in the matrix. Released into the water, the cloud spreads and hangs, a slowly expanding constellation of blue points, and it does not go out quickly. The observed persistence ran to several minutes, and in some cases close to 10. That is a very long time for a bioluminescent display. Most flashes in the sea last a second or less. Imagine the effect from the point of view of something that has just made contact with a vampire squid in complete darkness. A moment ago, there was one small target, detected by touch or by the faint disturbance of water. Now there is a spreading field of blue light, several times the size of the animal, drifting, glittering, going nowhere, occupying the exact place where the target was. Somewhere inside or behind or beyond that cloud, a red-brown animal that reflects no blue light whatsoever has folded its arms, dimmed its photophores, and begun beating its fins slowly away into the dark. It is the same idea as ink, executed with the opposite substance, because in that environment the thing that obscures is not darkness but light. Bioluminescence itself is worth a moment, since the sea runs on it and hardly anybody ever sees any of it. The chemistry, in nearly all cases, involves a small molecule generically called luciferin and an enzyme called luciferase. The enzyme brings the luciferin together with oxygen, the molecule is oxidized, and the energy released comes out not as heat but as a photon. That is the whole trick. It is a chemical reaction whose product is light, and it is remarkably efficient, losing very little of its energy to warmth. There is a quiet irony in that, given where our animal lives. Light production requires oxygen, and the vampire squid inhabits the least oxygenated water in the open ocean. It is spending, on its one flamboyant act, a resource it has otherwise organized its entire body around conserving, which tells you the display is worth a great deal to it. The light produced is blue, as almost all marine bioluminescence is, centered in the wavelengths that travel furthest through seawater, since red light would be absorbed within a few meters of wherever it was made, while blue carries on into the distance. It is difficult to convey how much of this there is in the ocean, because it happens almost entirely in places where nobody is looking. Surveys made with sensitive instruments lowered into the middle water suggest that a very large proportion of the animals living there are capable of producing light, and in some layers and some groups the figure approaches the whole community. Fish do it, using organs along the belly and beneath the eye, and in a few remarkable cases using bacteria that they farm in pouches. Shrimp do it, spitting clouds of luminous fluid in the same way our animal does. Jellyfish do it, running waves of light along their bells. Siphonophores do it, and some of them use their lights as lures on the end of extended tentacles. Squid do it, sometimes with organs of a complexity that includes lenses, reflectors, and color filters, arranged along the underside with such precision that the animal can match the faint light coming down from above and cast no shadow to anything hunting from below. There are single-celled organisms in the surface water that flash when the water around them is disturbed, and if you have ever swum at night in the right place at the right time of year and watched blue sparks come off your own hands, you have seen it. That is the same chemistry, in the same ocean, 400 meters shallower. Taken together, it means that light in the sea, below the reach of the sun, is almost entirely a biological product. It is made by animals, for the purposes of animals, in brief local events that begin and end in seconds. There is no ambient illumination down there at all. There is only what somebody switches on. So when a vampire squid ejects a slow-spreading cloud of luminous mucus that hangs in the water for minutes, it is not adding a small light to a lit place. It is creating, briefly, the only thing visible for a very long way in any direction, and then quietly stepping out of it. And the vampire squid does not only glow from its arm tips, its skin is scattered with photophores, small light organs, in the hundreds, distributed across the mantle and the head and the outer surfaces of the arms and web. They are not all identical, and they are not all doing the same job. And honestly, we do not fully know what all of them are for. The two large organs set behind the fins on the upper surface of the mantle are the most conspicuous. They are pale and rounded, and considerably bigger than the rest, and they have lids. A fold of tissue can be drawn across each one, closing it off completely, so that the animal can go from lit to unlit at will. Observers have watched this happen. An animal with those organs glowing will, without any other apparent change, simply switch them off and become invisible again. What the large organs are used for is not settled, They have been described as looking like a second pair of eyes, and a superficially eye-like pattern at the rear of an animal is a known trick elsewhere in nature. Though whether that is what is happening here is speculation, they may function in signaling to other vampire squid. In a place where finding another of your own kind is a considerable problem. They may serve to startle. They may do several things at once. The honest position is that we have watched them being turned on and off, and we do not yet know what the animal means by it. The smaller photophores scattered over the body have been suggested to play a role in counter-illumination, which is the practice of producing a faint glow on your underside to match the dim light filtering down from above so that you cast no silhouette to anything looking up. Many midwater animals do this, and do it with great precision. Whether the vampire squid does is questionable, because at 600 to 900 meters in a productive tropical region there is essentially no downwelling light left to match. The case is stronger for the shallower part of its range than the deeper part, all of which is a way of saying that this animal, which does almost nothing, is nonetheless equipped with a lighting system of considerable subtlety, and we can describe its parts far better than we can explain them. There is one more thing it does, and it is the most photographed behavior in its repertoire. When a vampire squid is disturbed persistently, or handled, or approached too closely by a vehicle, it will sometimes fold itself inside out. The 8 arms are swept backwards and upwards over the head and the mantle, and the web between them, which normally hangs beneath the animal like a shallow bowl, is turned over the top of the body and drawn tight. The result is a rounded shape enclosed entirely in the web, with the outer surface of the arms now on the inside and the inner surface, the surface bearing the cirri, now facing outwards in every direction. From outside, it is a dark bell studded all over with soft projections, and the animal has become, effectively, a closed object with nothing exposed. The eyes are covered. The mouth is deep inside. There is no soft edge to take hold of, and nothing to pull. It is usually called the pineapple posture, occasionally the pumpkin posture, and it is what nearly every well-known photograph of this animal is actually showing. It is worth knowing that, because it means the image that has settled in the public mind is an image of a frightened animal in a defensive ball, photographed by someone who had just alarmed it. It is a bit like assuming a hedgehog is normally a sphere. In its ordinary life, undisturbed, at depth, it does not look like that at all. It looks like a small, dark shape hanging level in the water with its web relaxed and a single thread out into the night. There is a sensible logic behind the posture. Combined with the luminous cloud, it gives the animal a 2-stage response that costs almost nothing. Stage 1. Release a spreading field of light where you were and move slowly away in the dark. Stage 2. If that fails and contact persists, close up into a sphere with the softest parts on the inside and wait it out. There is no sprint in it, no struggle, and no expenditure the animal cannot afford. For an animal with the muscle tone of a jellyfish and the oxygen budget of a sea anemone, going still and going dark is very close to the only option available, and it has been refined into something rather elegant. Almost every squid and octopus alive has the same shape of life, and it is a shape that surprises people when they first hear it. They grow extremely fast. A common octopus can go from a hatchling the size of a grain of rice to a full-grown adult in a year or a little more. Many squid do it in less. They eat enormously, convert food to body tissue with an efficiency that is unusual among animals, and they reach maturity while still, by the standards of a vertebrate of similar size, very young, and then they breed once, and they die. The technical word is semelparity, breeding a single time, and in the cephalopods it is not a matter of exhaustion or bad luck. It is programmed. In the female octopus, the process is controlled by a pair of glands behind the eyes, and once the eggs are laid, a hormonal cascade begins that shuts the animal down. She stops feeding. She stays with the eggs, cleaning them and passing water over them, for weeks or in some deep-sea species for a very long time indeed, and she does not eat during any of it. When the eggs hatch, she is spent, and she dies shortly afterwards. The males die too, some weeks or months after mating, by a similar route. It looks tragic when it is described in those terms, but from the point of view of the lineage, it is perfectly sound arithmetic. If you can grow fast, and if you can produce a very large number of eggs in one enormous effort, and if the environment you live in is rich enough to support that growth, then pouring everything into a single reproductive event and holding nothing back is the strategy that produces the most offspring. Anything kept in reserve is wasted. That whole calculation depends on being able to grow fast, which depends on being able to eat a great deal of high-quality food. The vampire squid cannot do any of that. Its food is marine snow, which is thin material, largely already digested by something else, and it collects that food one filament load at a time by waiting for it to arrive. Its metabolism runs at a rate that would be described in any other cephalopod as pathological. It has no way of accumulating the enormous energy surplus that a single terminal spawning would require. If it tried to follow the standard cephalopod strategy, it would take an extremely long time to build one clutch, and it would then have precisely one chance for that clutch to work, in an ocean where the survival of any given batch of eggs is close to a lottery. So it does something else, and this was not established until 2015. A group including Hoving, working then at the research center in Kiel, together with colleagues in Britain and the United States, went to a museum collection of vampire squid taken off Southern California over a period of decades. They looked at the ovaries and the reproductive tracts of some 40 females at a range of sizes and stages, and they looked specifically for the anatomical traces that spawning leaves behind. When an egg is released from an ovary, it leaves a mark. The follicle that held it collapses and is reabsorbed, and for a while there is a visible structure where it was. Count those, and you have a record of how many eggs have already gone. What they found in some of the specimens was a combination that should not have existed. There were females whose ovaries showed clear evidence of having released eggs, in quantity, and which at the same time contained large numbers of new eggs at an early stage of development, and which were in every other respect in good condition. They were not spent. They were not shutting down. They had spawned and then gone back to a resting state and begun again. In at least one individual, the traces suggested a very large number of separate spawning events, running into the dozens, with thousands of undeveloped eggs still held in reserve. That makes the vampire squid, so far as anyone knows, the only living coleoid cephalopod that does not follow the breed once and die pattern. It spawns in batches, rests, rebuilds, and spawns again, over and over, across a life that must therefore be considerably longer than a squid's. How much longer is not directly known, and this is a place to be honest about the limits. Nobody has kept a vampire squid alive in a tank for anything like its natural lifespan, and nobody has followed an individual in the wild. What can be done is to look at the hard parts, the beak and the mineral grains in the statocyst, which in many cephalopods lay down growth increments in something like a regular rhythm, and to read them the way you would read tree rings. That work has been attempted for this species and for its deep-living neighbors, and the results point in the direction you would expect from an animal with this metabolism, which is towards a life measured in years rather than months, and quite possibly a good many years. Estimates in the region of 8 years have been put forward. They should be held loosely. There is a further detail from the reproductive work that is quietly touching. There is evidence that a female, if conditions are poor and the food is not arriving, can reabsorb her own developing eggs, taking the material back into her body rather than losing it. She is not committed. She can wait and try again later, in a way that no other soft-bodied cephalopod can. The eggs themselves are large by cephalopod standards and few in number in any given batch, and as far as anyone can tell, they are released singly into the open water rather than laid in a mass or attached to anything or brooded. They drift. Development is presumably slow, since everything about this animal is slow and the water is cold, though nobody has watched an egg through to hatching. Hatchlings have been caught occasionally, and they are tiny, a few millimeters, transparent, with a pair of fins near the front of the mantle and arms not yet joined by much of a web. There is some evidence that the very young live higher in the water column than the adults do, in better oxygenated water, and work their way down as they grow and as their physiology becomes capable of the low oxygen layer. That would make a good deal of sense. A hatchling has a very high surface area for its volume and cannot possibly carry the metabolic machinery of the adult, and the deep layer would be closed to it, and the mating itself has never been seen. Not once, in more than a century. In most cephalopods, the male has a specially modified arm, the hectocotylus, used to transfer packets of sperm to the female. The vampire squid male does not have one. Females have been found with sperm packets implanted in the tissue around the head and the base of the arms, so we know the transfer happens and roughly where it lands. But the act itself, in the dark, at 800 meters, between two soft animals that meet each other only rarely, has never been observed by anyone. That is one of a good many things about this creature that we simply have not seen yet. It would be wrong to leave the impression that the vampire squid hangs there alone. The oxygen minimum zone is thinly populated compared with the water above it, but it is not empty. and the animals that do make a living there are worth knowing about, because they are all working versions of the same problem from different angles. The most numerous vertebrate on the planet lives in this general layer, and almost nobody has heard of it. It is a small fish of the genus Cyclothone, one of the bristlemouths, and it is perhaps 2 or 3 centimeters long, thin, soft, with a large mouth full of fine teeth, and a body so lightly built that it barely holds together in a net. There are estimated to be hundreds of trillions of them, possibly more, spread through the twilight water of every ocean. They do not migrate much. They hang in the dark and wait for small things to come near, and they get by on very little. If you were ranking animals by how much of the Earth's vertebrate biomass they represent, Cyclothone would embarrass every creature you have ever seen in a documentary. There is a large red shrimp, sometimes called the giant red mysid, which was for many years the standard laboratory subject for anyone studying life at low oxygen. It lives in and around the same layer off California, and it turned out to have a respiratory pigment with an extremely high oxygen affinity, together with a very efficient ventilation system, and the work done on it in the 1970s and 80s provided the framework that people later applied to the vampire squid. There are other shrimp in the layer that have adopted a different tactic entirely, which is to stop. Researchers using the Monterey vehicles documented pelagic shrimp in the core of the low-oxygen water going completely limp, hanging motionless with their limbs slack, in a state that looks from outside like death, and appears in fact to be a deliberate metabolic shutdown. They ride out the worst of the layer in suspension and resume when conditions allow. There is a small, transparent octopus called Japetella, which shares a good deal with our animal, including a gelatinous body, minimal muscle, and one of the lowest metabolic rates in its own group. It lives in overlapping water and is subject to the same constraints and has arrived at a broadly similar answer, though it remains a predator in the ordinary sense. And there are the gelatinous animals, which are everywhere in the deep and which are systematically underrepresented in the older literature because they do not survive nets. Cephalopod, which are colonies of specialized individuals strung out along a shared stem, some of them tens of meters long, drifting with curtains of stinging tentacles hanging beneath them. Comb jellies rowing along on rows of beating hairs that throw rainbows in a submersible's lights. The giant larvaceans in their houses of mucus, a meter across in some species, filtering the water and building and abandoning and building again. Above all of it, twice a day, the Great Migration passes through. Lanternfish in their millions, rising at dusk and sinking at dawn, and krill, and squid, and the copepods, and behind them the animals that eat them. That traffic is the reason the snow keeps falling. Every one of those animals that dies at depth, and every fecal pellet any of them produce, feeds the layer below. The vampire squid, being what it is, takes no part in any of this. It does not migrate. It does not chase the traffic. It stays where it is and collects the residue. Does anything eat it? The answer appears to be yes, but not often, and the evidence is indirect. Vampire squid remains have turned up in the stomach contents of large, deep-diving fishes and of some marine mammals capable of working at those depths. Beaks are the usual find, because a cephalopod beak is made of hardened chitin and it survives digestion when everything else is gone, and beaks accumulate in the stomachs of animals that eat a lot of cephalopods. But the numbers are modest, and the reason is the same reason the vampire squid is there in the first place. To eat one, you have to get to it, and getting to it means entering water where you cannot sustain yourself. A deep diving whale can do it on a breath hold, briefly, on a dive it is planned. A resident predator cannot, because there is no such thing as a resident predator in that layer. The oxygen minimum is a wall, and the vampire squid lives on the far side of it. Smaller things get through, though. In recent years, the Monterey vehicles have recorded midwater cephalopods, including this one, carrying parasites on their gills, visible on video as pale attached forms clinging inside the mantle cavity. Parasitic copepods and their relatives have found a way in that a fish cannot, by arriving as tiny larvae drifting in the water and simply latching on. Life gets everywhere. Even a habitat defined by the near absence of oxygen has its passengers. There is one more thing worth mentioning about this layer, and it needs saying carefully, without alarm, because it is a real feature of the present ocean. Oxygen minimum zones are not fixed. They respond to how warm the surface water is, because warm water holds less dissolved gas, and because a warm surface layer sits more stubbornly on top of the cold water beneath it, and mixes with it less readily. Both of those effects reduce the amount of oxygen getting down. Measurements taken over the last several decades indicate that the low oxygen regions of the world's oceans have been growing. spreading outward at their edges and rising slightly towards the surface. For most animals in the middle water, that means a habitat squeezed from below. The vertical migrators that shelter at depth by day and feed at the surface by night have less room to work with, and the compression of that range is something biologists are watching closely. For the vampire squid, the arithmetic runs the other way. Its habitat is defined by the low oxygen layer, and if the layer expands, so, at least in principle, does its range. Whether that translates into more vampire squid depends on a great many other things. Chief among them, whether the snow keeps falling in the same quantity from the surface above. It is not a simple story, and nobody should pretend it is. But it is at least possible that among the winners of a changing Middle Ocean will be a soft red animal that gave up hunting a very long time ago in exchange for a room nobody else wanted. On the 13th of September, in 2016, a research vessel working off Hainan Island in the northwestern South China Sea put a net down to 1,000 meters and hauled it slowly upward. It was not a hunt for anything in particular. It was an open cruise, the routine sampling work that keeps an oceanographic institute supplied with material, and the net in use was a multi-net, a rig carrying several separate nets that can be opened and closed in sequence, so that each one samples a defined band of depth. It came up through a series of layers, closing one net and opening the next, and among the contents of the deepest band, the water between 800 and 1,000 meters, there was a single adult female vampire squid. That is worth noting in itself. The genus had not previously been recorded from the South China Sea at all. It was a first for the region. The specimen went back to the South China Sea Institute of Oceanology and was photographed, measured, and described, and then it sat, as specimens do, while the work of comparison was done. And when it was compared, in detail, with the standard description of Vampyroteuthis infernalis, 3 differences kept coming back. The first was at the rear end of the mantle. The specimen tapered to a distinct point, a small acute tail, and the standard description of the vampire squid does not include one. The second was the position of the pair of large photophores. In the usual description, those organs sit about 1/3 of the way along the distance between the fins and the rear end of the body. On this animal, they were at the midpoint. The third was in the beak. Cephalopod beaks are made of hardened chitin, and their shape is one of the more reliable identification characters available, precisely because they survive when everything else has softened. The lower beak of this specimen had a wing, a lateral flange, that was broad and noticeably elongated, where the described form has a broad but short one. To that, the team added molecular work, sequencing a mitochondrial gene commonly used for species identification, along with sections of ribosomal DNA, and building trees comparing their animal with sequences from vampire squid collected elsewhere in the world. They concluded that they had a second species, and in 2024 they published it, naming it Vampyroteuthis pseudo-infernalis. If it holds, it is the first addition to that order of animals in 121 years. An order that had contained exactly 1 living member since 1903 would contain 2. The reception has been mixed, and that is entirely normal, and it is worth understanding why, rather than treating it as a controversy. Describing a new species from a single specimen is legitimate, and it happens often, particularly in the deep sea, where a single specimen may be all anyone gets in a decade, but it carries an unavoidable weakness, which is that you cannot tell the difference between a character that distinguishes a species and a character that distinguishes an individual. Animals vary, populations vary. Without a series, you have no way of knowing which of the differences in front of you are meaningful. There is a second weakness, and readers who have been paying attention will have seen it coming. The specimen was collected in a net, towed vertically for a considerable distance, and this is an extremely soft animal. A small acute tail at the rear of a gelatinous mantle is precisely the sort of feature that a net can create or destroy. Tissue stretches, tissue tears. The rear tip of a soft mantle drawn through mesh and then preserved may take a shape it never had in life. And there is the long shadow of the century we have already been through, in which 11 species in 8 genera were erected on exactly this kind of evidence, and then collapsed into one by somebody who finally had enough specimens to lay out in a row. More recently, another group went back over the question. They re-examined vampire squid specimens from the Gulf of Mexico, which the original study had suggested might belong to the proposed new species. They looked for the acute tail and the elongated beak wing and did not find them. They took the original genetic dataset and reran the analyses, then built new trees, adding a further gene and combining the markers together, and none of the resulting trees separated the proposed species from Vampyroteuthis infernalis in a way that supported a split. Their conclusion was that the name should be treated as a synonym, and that what is being seen is variation within a single widespread species. So where does that leave it? Genuinely unresolved, and comfortably so. The proposal is on the record and available for testing. The reanalysis is on the record too. Many working cephalopod specialists continue to treat the vampire squid as one species until more material arrives, which is the conservative position and probably the right one to hold for now. Others think the variation across the world's oceans is real, and that this will eventually be resolved into more than one animal, which would not be surprising for a creature distributed across every warm ocean on Earth in a habitat that is patchy and discontinuous. What would settle it is more specimens, a series from the South China Sea at a range of sizes. Fresh tissue for sequencing, collected carefully rather than in a towed net. Ideally, footage of living animals in that region, so that the shape of the mantle can be seen in an animal that has not been dragged through half a kilometer of water. All of that is achievable. It simply has not been done yet, which is a fair description of the state of knowledge about this animal in general. It is worth spending a little time, before we go any further, simply trying to sit inside that life. Not to claim we know what it is like. We do not, and there are good reasons to be careful about the whole exercise, but we can list what the senses of this animal report and what they do not, and from that we can build something like a picture of the world it occupies. Begin with weight, because that is where the difference from your world is largest. You have never in your life been free of your own weight. Even lying down now, entirely still, your body is pressed against the mattress and the mattress presses back, and there are muscles in your neck and your back that have never fully switched off. You know which way is down because you are being pulled that way continuously and have been since the moment you were born. The vampire squid has no weight. Its tissues are so nearly the density of the water around it that gravity, for practical purposes, has been cancelled. There is no down. There is no surface pressing back. There is no part of the body that is bearing the load of another part. It is suspended entirely and equally, on every side, in a medium that supports it perfectly. The only reason it knows its orientation at all is that a few dense grains resting on sensory hairs inside its head continue to report which way is which. Take that organ away, and there would be no up in its world whatsoever. Now, the pressure, which is the other side of the same coin. At 800 meters, the water is pressing in at something like 80 times the pressure at the surface. That sounds crushing, and it is not, because the animal is made almost entirely of water, and water does not compress. The pressure inside equals the pressure outside at every point. There is nothing to crush. The animal does not feel it any more than you feel the weight of the atmosphere sitting on your shoulders right now, which is considerable and which you have never once noticed. The temperature is around 4 degrees, and it does not change. Not by day, not by night, not with the seasons. Whatever the weather is doing, 800 meters above, whatever the storms are doing to the surface, none of it reaches down here. This is water that has not seen the sky for a long time. The light. There is, at that depth in a productive tropical ocean, effectively no sunlight. Perhaps at the shallow end of the animal's range, on a clear day, in the clearest water, there is the faintest residue of blue overhead, enough for a very sensitive eye to register a difference between above and below. Deeper than that, nothing. But it is not a dark world in the way a sealed room is dark. It is a dark world with lights in it. Bioluminescence in the deep sea is not rare. It is close to universal. Estimates vary, and the sampling is difficult, but a large fraction of the animals living in the middle water can produce light, and they do so constantly, for reasons ranging from attracting mates to startling predators to lighting their own way. So what the vampire squid actually sees, through those enormous light-gathering eyes, is a black field across which points and flashes appear. A blue spark, far off, gone. A slow drifting glow that might be a siphonophore's line of lights. A sudden burst somewhere below when something is disturbed. A long streak as something moves. There is no scene, no landscape, no continuous image of a world. There is a darkness containing occasional events, and the enormous eyes exist to make sure that none of those events go unnoticed. Then there is touch, and touch is where this animal actually lives. The filament is the primary interface between the vampire squid and its surroundings, and it is a very strange kind of interface. It is a single line, extended out into a volume, reporting contact. Everything the animal learns about the falling snow, It learns through that thread. Something arrives, or it does not. There is no image of the food, no smell of it in the ordinary sense, no sight of it approaching. There is a length of nerve held out into the dark, and at intervals, the news that something has landed. Beyond the filament, the arms and the cirri and the web are all sensitive. Cephalopod skin generally is well supplied with receptors, and cephalopods also carry lines of specialized cells along the head and arms that detect movements of water in something like the way a fish's lateral line does. So the animal has a sense of the water around it, of disturbance in it, of something passing nearby without touching. In a place with no visibility, that matters a great deal, and there is chemistry. Cephalopods taste with their arms, and the vampire squid presumably samples what its filament brings in and what its web encloses. Sound is a strange one to think about down there. The deep ocean is not quiet. Sound travels through water 4 times faster than through air, and it travels enormous distances, particularly in a layer around 1,000 meters where the physics of temperature and pressure conspire to trap it and channel it, so that a low-frequency call made in one ocean basin can, in principle, be detected on the other side of it. The calls of large whales are in the water constantly. So is the rumble of distant storms and the grinding of the seafloor, and in the modern ocean, a great deal of shipping. Cephalopods do not hear in the way we do, having no gas-filled ear to be moved by pressure waves. What they have is the statocyst, and the statocyst can detect the particle motion component of low-frequency sound. So it is probably fair to say that a vampire squid does not hear the ocean exactly, but is moved by it, very slightly, in a way it can detect. Put all that together and you have the following existence: weightless, in permanent darkness, at a constant cold temperature, in water that never moves quickly, with no floor and no ceiling and no direction except the one your balance organ tells you about. A world without weather, without seasons, without day or night in any meaningful way, except that twice every 24 hours a vast quantity of small animals passes overhead, going up and later coming down, and their traffic and their leavings are the closest thing this place has to a calendar. And through all of it, continuously and without pause, the slow fall of everything from above, an animal that hangs level in that, entirely still, for hours at a time, with one thread out. It is very difficult to think of a life on this planet that involves less hurry. Here is a list of things we do not know about this animal, offered plainly, because the gaps are as interesting as the findings, and because a documentary that pretends otherwise is not worth much. We do not know how they mate. The event has never been witnessed. We know it happens because females carry the evidence of it, and we know roughly where on the body the transfer lands. The rest is inference. We do not know how they find each other. 2 soft animals, a foot long, drifting in a layer of water that spans entire ocean basins, at densities so low that a vehicle can search for hours without finding one. Something must bring them together. Chemical trails have been suggested. The large photophores may be involved, on the reasonable grounds that a light you can switch on and off is a signal waiting to happen. Nobody knows. We do not know what happens to an egg. None has been followed from release to hatching. We do not know how long development takes, or what depth the eggs drift at, or what proportion of them survive. We do not know the lifespan. The estimates come from counting increments in hard structures and from reasoning about metabolic rate, and they point towards years rather than months, and possibly towards a good many years, and that is genuinely as far as it goes. We do not know how many there are. There is no reliable abundance estimate for vampire squid anywhere in the world. You cannot survey them with nets, because nets destroy them and because they are patchily distributed. You cannot easily survey them acoustically, because they have no gas-filled structures to return an echo. The only reliable counting method is the one that requires a ship, a vehicle, a cable, and a full day of somebody's time for each sighting. We do not know how far a filament can actually extend in the wild. The figures quoted vary a great deal, and the reason is that measuring a fine, transparent thread against a black background at 800 meters from a moving vehicle with lights that only illuminate a limited volume is extremely difficult. It is considerably longer than the animal. Beyond that, the honest answer is that it is hard to say. We do not know how many species there are, as we have just seen. We do not know a great deal about the nervous system. The octopus has been studied intensively for decades, its behavior and its learning, and the remarkable distribution of its neurons through its arms. The vampire squid has not, for the straightforward reason that you cannot keep one. Animals brought to the surface survive a matter of hours, or at best days in a chilled tank, long enough for a feeding experiment and not remotely long enough for anything else. Everything we would like to ask about how this creature perceives and decides sits behind that wall, and underneath all of those specific gaps is a much larger one. The middle water of the ocean is the largest living space on this planet. Not the largest habitat on land, not the largest habitat in the sea, the largest anywhere, by an enormous margin, when you measure it the way it should be measured, by volume. The continents are surfaces. Life on land occupies a thin film a few tens of meters thick, from the deepest roots to the tallest canopy. The open ocean is a volume kilometers deep, spread across most of the planet, and the twilight middle of it, the part we have been in tonight, accounts for a staggering share of the total, and the fraction of that volume that any human being has ever seen, directly or through a camera, is so small that it does not usefully round to a number. All the submersible dives ever made, all the remotely operated vehicle hours, all the towed camera transects, added together, come to a set of thin threads drawn through an unimaginably large space. Most of what lives out there has never been observed alive. A great deal of it has never been observed at all. That is the context in which the story we have been telling actually sits. The vampire squid is not an obscure animal. It is one of the best studied creatures of the deep middle water, the subject of a monograph tradition going back a century, a museum specimen in every major collection, a name known to anybody with a passing interest in the sea. It has been filmed hundreds of times by one of the best equipped ocean research institutes in the world, in a canyon, a short boat ride from the shore of California, and it took until 2012 to establish what it eats. Whatever is going on out there in the parts nobody has visited, and there is a very great deal of it going on, we have not begun to see it. That is not a gloomy thought, and it is not meant to be. It is simply the size of the thing. We are talking about a world that occupies most of the volume of our own planet, that runs on a currency of falling remains, that is lit almost entirely by its own inhabitants, and that has been operating in exactly this way, with variations, for hundreds of millions of years. We have been sending down instruments for about 150. It would be strange if we had finished. Let us go back, one last time, to a tired man in the hold of a converted cargo steamer, somewhere in the tropical Atlantic in the winter of 1898, with a small dark animal in a tray in front of him. It is easy, from here, to be unfair to Carl Chun. He gave this creature a name it did not deserve, and the name shaped everything that was thought about it for a century. And the temptation is to treat that as a failure of judgment. It was not, particularly. He had a soft, damaged animal, dark red, webbed, with large reddish eyes, brought up from a depth nobody had any real feeling for, in a period when the deep sea was less a place than a rumor. He described it accurately. He noted that it was gelatinous and probably inactive, and he was right about both, and it took 80 years for anyone to demonstrate how right. He simply also gave it a name that was a piece of 19th century Gothic scene setting in a scientific culture where that was ordinary, and he had no way of knowing that the name would travel further and last longer than any of the careful observations attached to it. Science is full of these. The horseshoe crab is not a crab and is closer kin to spiders. The Portuguese man of war is not one animal but a colony of many. The electric eel is not an eel. The koala is not a bear. The flying fox is not a fox. And the mountain chicken is a frog. Names stick with a tenacity that has nothing to do with their accuracy. Because a name is not a description, it is a handle, and once enough people are holding it, nobody can change it. But there is something particular about this case, because the name did not merely mislabel the animal. It described a way of living, and it described the opposite of the truth. What the vampire squid actually is a gleaner. The word is an old agricultural one. Gleaning is what was done in a harvested field after the reapers had passed, by people who came behind and gathered what was left on the ground. It was not hunting, and it was not farming. It was the patient collection of what the main effort had finished with, and in most old agricultural societies it was protected by custom or law, on the grounds that a field should not be stripped so thoroughly that nothing remained for those who came after. That is exactly what this animal does. The great harvest happens in the sunlight at the top of the sea, in the bloom and the grazing and the enormous nightly migration, and it is loud and fast and competitive, and it consumes almost everything it produces. And what falls out of the bottom of it, the fraction that escapes, the crumbs and the leavings and the discarded houses and the remains of the remains, drifts down for weeks into the dark. And there, at the edge of the layer where nothing else can afford to wait, is one small red animal, holding out a thread. There is a role in this that is easy to overlook, and it is not sentimental, it is ecological. Marine snow at 800 meters is fine, dispersed and low in energy, and there is almost nothing at that size in the middle ocean that can use it. The particles are too small and too scattered for a fish to bother with. Left alone, most of them continue downward or dissolve, and the energy in them goes to bacteria. The vampire squid concentrates it. It takes an enormously diffuse resource, spread through a huge volume of water, and gathers it, slowly, over a long life, into a body about the size of your hand. That body is then a coherent parcel of food, large enough for a deep-diving fish or a whale to be interested in a layer of water that would otherwise offer them almost nothing. And whatever the animal does not use, it releases as fecal pellets, which are denser and more compact than what came in, and which sink faster, carrying carbon further down than it would otherwise have traveled. So the animal is a bridge. It connects the finest, most dispersed material in the ocean to the largest predators in it, and it helps move carbon from the surface of the world into the deep, and it does all of that while spending less energy than almost anything else of comparable size and complexity anywhere on the planet. That is the animal that has spent 120 years being drawn with its arms flared and its cirri rendered as teeth. There is a lesson in there about names, and there is possibly a larger one about how quick we are to read appetite into an unfamiliar shape. But there is also something simpler, and I think it is the thing worth taking away tonight. The vampire squid is the last surviving member of an entire order of animals. Everything else on its branch is gone, and the fossils tell us that its ancestors were active, capable hunters who did what cephalopods have always done. Somewhere along the way, one lineage in that branch stopped competing. It stopped chasing. It gave up muscle, gave up speed, gave up the whole expensive apparatus of pursuit, and moved into the one part of the ocean that nothing else would take, and made a living out of waiting. Every one of its faster relatives is extinct. It is still here. So let us go back down there one more time, now that we know what we are looking at, and simply stay a while. It is late afternoon at the surface, somewhere over the eastern tropical Pacific, and there is nothing to see in any direction but water. The swell is long and slow, and there is no wind to speak of. The light is beginning to go gold at the edges, sink through the warm top 10 meters, where the light still moves in nets across everything, through 50, where the reds have already left and the world has begun to simplify, through 100, into the deep blue that photographs never quite capture, where you can still read by the light if you were somehow able to hold a book, through 200, where the last of the useful sunlight goes, and the water changes from a place with a color to a place with only a direction. Keep going. 300, 400. The temperature has been dropping all the way and is now in single figures. The pressure is building steadily and evenly, and there is nothing about it that is dramatic. Somewhere in here, hanging in the dimness, are the daytime layers of small fish and shrimp, the ones that will rise at dusk. There are so many of them that ships detect them as a false seafloor on their echo sounders, a phantom bottom that rises every evening and sinks every morning, and which puzzled naval sonar operators for years before anybody worked out that it was alive. 500. The oxygen is beginning to fall away. The water here has not touched the atmosphere in a long time, and everything that fell through it has been slowly burning up whatever it had. 600. We have arrived. It is entirely dark, and not merely dim, but dark in the way a cave is dark, in the way a closed room at night is dark. And the darkness goes on in every direction for a very long way. The water is around 4 or 5 degrees, and it is barely moving. There is no sound that you would recognize as sound, and the oxygen in this water is at a small fraction of what it is at the surface, low enough that a fast fish coming down here would find its own muscles turning against it within minutes. And it is snowing, slowly, continuously, in every direction, all the way down. Pale flakes and flecks and soft, ragged clumps, turning as they fall. Some of them the abandoned mucus houses of animals a few centimeters long. Some of them the packaged waste of creatures too small to see. Some of them the disassembled bodies of things that were alive in the sunlight 2 weeks ago and a kilometer above. Nothing about it is fast. Nothing about it stops. Hanging level in the middle of it, motionless, is a small red-brown animal about the length of a forearm. Its fins are not beating. Its arms are relaxed, the web hanging in a soft, open bowl beneath it. The enormous eyes are taking in a field of black with, very occasionally, a distant blue spark somewhere in it. The photophores on its skin are dark, It weighs nothing. It is holding its position perfectly without doing anything at all, and it has been doing so for some hours. From one side of the arm crown, a single fine pale thread runs out into the dark, far longer than the animal itself, and disappears beyond the edge of anything that could be called visible. Something touches the thread. The response is unhurried. The filament begins to draw back in, gathering, and as it passes through the arms, the suckers along them release mucus that strips what has arrived and binds it into a soft, small mass. The cirri, those rows of soft fingers, work it inward, hand over hand, down the arms to the mouth. The whole business takes a while, and there is nothing about it that resembles a struggle. Then the filament goes back out. Above, at the surface, the sun has set, and the great migration has begun. Uncountable small animals are rising through the water column, hundreds of meters, up into the dark surface to feed under the cover of night. They pass above this layer and do not enter it. In 8 or 9 hours, they will come back down, and behind them will come everything they have shed. The animal does not participate and does not appear to notice. Time passes differently down here, with the heart slow and the chemistry slower still. The food, once it is inside, will last a long while, because there is almost nothing burning it. A day in the life of an animal running at this rate is not the same length of experience as a day in yours, and there is no particular reason to think it feels long or feels short. It simply goes. The night passes above, dawn comes at the surface, and the migrating layers sink back into the twilight, and the snow that they and everything else have produced begins its long fall down, and in 10 or 14 days, some of it will drift past a small red animal that has not moved. It might do this for years. Somewhere in those years, it will find another of its own kind, in a way nobody has ever seen, and afterwards it will release a batch of eggs into the water, a few at a time, and let them go, and then it will rest and rebuild, and do it again, and again, dozens of times perhaps, across a life spent almost entirely still, and when it finally stops, it will begin to sink, slowly, the way everything does, down out of its layer and into the deeper water, and eventually onto the fine, pale ooze of the floor, where it will become part of the same falling that fed it. If you take nothing else from tonight, there are perhaps 4 things worth keeping, and they are all quite small. The first is that there is a layer of the ocean, hundreds of meters thick and spanning entire basins, where the oxygen has been drawn down so far that almost nothing with muscle can stay, and that this same layer, for exactly the same reason, is the one with the most food falling through it. The barrier and the abundance are 2 faces of a single process. That is a lovely piece of natural bookkeeping, and it explains almost everything about the animal we have been following. The second is that this animal got in by giving things up. It gave up speed, gave up muscle, gave up the pursuit of anything, and lowered its energy requirements below the level that the oxygen imposes, and in return it received an entire habitat with no competition and very few predators, and a food supply that never stops arriving and never has to be caught. It is one of the clearest examples in nature of a lineage winning by asking for less. The third is that the deep sea keeps its habits well hidden, and that a name given in a hurry can stand in for 100 years of knowledge. Chun described the animal correctly and named it wrongly, and it took submersible robots, a canyon in California, 20 years of video, and 4 separate lines of evidence to establish something as basic as its diet. There are a great many animals down there about which we know considerably less, and some of them are almost certainly doing things at least as surprising. And the fourth is simply this. The thing that finally explained the vampire squid was not a bigger net or a stronger light or a deeper machine. It was patience. Somebody went down and watched, and kept watching, over 2 decades, until the animal did something ordinary in front of a camera, which is a fitting way to learn about a creature whose entire strategy is to hold still and wait. It is getting late now, so let us go back up, slowly, and then let the whole thing go. Come up off the layer first. Leave the small red animal where it is, hanging level in the dark with its thread out, because it will still be there and it is in no hurry and there is nothing that needs doing. Rise through 800 meters, and then 700, and then 6. The snow is falling past you the other way as you go, all of it drifting down, unhurried, turning as it falls. You are moving up through weeks of it. Every flake you pass came apart at the surface at some point in the last month, and has been on its way down ever since. At 500, the oxygen begins to come back into the water, a little at a time. At 400, there are the daytime layers, the small fish and the shrimp waiting out the light. At 300, there is the faintest suggestion that there is such a thing as above. 200, and the blue begins. not color exactly. Direction, 150, and it is the blue of a very late evening. 100, and there is light enough to see your own hands. 50, and the surface is a moving ceiling of silver somewhere overhead, and the sun is coming through it in long, slow columns that swing as the swell passes, and then out into the air, on a calm morning, on a sea with no land anywhere on it, with the light coming up. Everything we have talked about tonight is still happening underneath that. It is happening right now, and it will still be happening while you sleep. In the Eastern Pacific and the Arabian Sea, and off the coast of Peru, and out beyond the canyon, in Monterey Bay, in a band of water 600 to 900 meters down, the snow is falling and the oxygen is thin, and there are small, soft red animals hanging in it with a single thread held out, waiting for something to arrive. They have been doing it for a very long time. Their lineage was already old when the last dinosaurs were alive. Everything else on their branch is gone, and they are still there, weighing nothing, needing almost nothing, gathering what falls. Now come back to the room, because the thing that started us off tonight is still true, and it is happening around you at this moment. There is dust falling in the air above you, very slowly, far too slowly to see. Fibers from the blanket, and small pieces of the day, drifting down through the dark of the room, past the edge of the bed, settling on the floor and the shelf and the back of your hand, and it does not stop when you close your eyes, and it will not stop while you sleep. And there is nothing at all that you need to do about it. It is the gentlest process there is. Nothing about it asks anything of you. You do not have to catch any of it. Let your shoulders come down from wherever they have been holding themselves. Let your jaw loosen. Let the weight of your head go properly into the pillow, so that your neck is not doing any work at all. And let your arms be heavy, and let your legs be heavy, and let the bed take all of it, the way the water takes the weight of an animal that has not carried itself in 180 million years. That is what that creature has, down there in the dark. Complete support on every side, with nothing to hold up and nothing pressing back, and no direction it has to move in and no reason to move in it. For the next little while, you can have some of that too. There is nothing you need to work out tonight. Nothing needs solving before morning. Whatever is waiting will still be waiting, and it will be easier, and it will not be improved by being turned over in the dark. You are allowed to put it down. You are allowed to be exactly as still as an animal that has made stillness its entire way of living. Let your breathing go slow and low, and let it find its own rhythm, without you managing it, in and out, the way the swell moves at the surface, long and unhurried, with a great deal of quiet in between. And if your mind wants something to hold, let it hold the falling. Let it be snow, if you like, the ordinary sort, going down past a streetlight on a windless night, in that soft, wandering way that snow has when there is no hurry in the air. Or let it be the other snow, the pale, slow rain of the deep ocean, drifting down through water that has not seen the sun in years, past nothing, towards nothing in particular, in no hurry whatsoever. And somewhere out in the middle of that, level in the dark, entirely still, weighing nothing at all, a small red animal with a thread held out into the night, waiting for the world above to send something down. It is not doing anything else. It does not need to. Neither do you. Let it go now. Let the room go soft at the edges, and let the sound of my voice become just another part of the dark, something drifting past rather than something to follow. And if you lose the thread of it, that is exactly right, and you should let it go. The sea will keep on doing what it does. The snow will keep falling in the middle water. The small red animal will hold its position through the night and through tomorrow and through the day after, patient and unhurried, gathering what falls, needing almost nothing. And you can sleep now. Sleep well. The sea keeps its slow watch in the dark, and it will still be turning, far below, long after the light is gone.